Hi everyone, welcome to my channel Pure Biology, and today I come with another video that is the DNA binding motifs, and this topic is related to the transcription process. That means here the binding proteins, how they bind with each other, that is related to this transcription process. So my any different topics related to the transcription process already explained in my channel, so you just check out that all. This is the DNA binding motifs, and what is this? The binding motif that permits the specific binding of DNA binding protein to its target sequence. That means it helps in the binding of DNA binding protein to its target sequence. They generally help in the binding process, simple way. And here are different types of motifs. These are discovered or these are invented by the scientists. And here in the first video, I generally try to explain that helix turn helix motif and helix loop helix motif. Another two important motifs, these are also related with this transcription process. I will explain in my next video. Okay. So this helix turn helix motif is what? This is present both in the eukaryotic and the prokaryotic DNA binding proteins, and it was the first DNA binding structure to be identified. Clear? The motif is made up of two alpha helices and separated by a beta turn. So here is the structure that is explained. Motif that is made up of the two alpha helices, and there is one beta turn that helps the two alpha helices to be separated from each other. And this beta turn that consisting of the 4 amino acids, and this turn in conjunction with the first alpha helix position the second alpha helix on the surface of the protein in an orientation that enables it to fit inside the major groove of a DNA molecule. So what is saying that this beta turn attach the two alpha helices in such a way like that. This is the rough diagram of this helix turn helix. So this is the beta turn and these two are the helices. Okay. So first one and second one, these are just separated by this beta turn in such a way that making a position and angle. You can say that first one and second one, they are attached to each other by the beta turn, forming an angle. And why this way they remain? Because in remaining in this position, it is very helpful to them, or not helpful, it just helps them to attach with the DNA very perfectly. So that's why it is saying here that the surface of the protein in an orientation that enables it to fit inside the major groove of the DNA molecule. So in that way they remained. That's why they can easily attach with the major groove of the DNA molecule. Okay. The second helix is known as the recognition helix. Here are the two helices. This is the first helix and this is the second helix. The second helix is known as the recognition helix. Okay. So this is the helix turn helix motif. And here, sorry, the homeodomain is an extended HTH motif that possessed by the proteins coded by the homeotic genes. So this is one kind of HTH motif that is present where? That is present in the homeotic genes, and it is made up of generally 60 amino acids and encoded by 180 BP DNA sequences and called the homeobox. So that means 60 amino acids present in this homeodomain. This is one kind of example you can say. And this is encoded by one DNA sequence that is 180 BP, and this is known as that homeo box. Okay, and it has 3 alpha helices. So generally here just we have learned the 2 alpha helices are present and these are separated by the beta turn, one beta turn. But in case of homeodomain, this consisting of the 3 alpha helices and these are separated by the obviously beta turn. And 2 and 3, generally 2 and 3, these are mainly separated by the beta turn. Not the first one, second and third one, that are separated by the beta turn. And helix 3, which is 17 amino acid residues, that means it's consisting of 17 amino acids and act as the recognition helix. So helix 3 act as the recognition helix and binds in where this binds with the major groove of the DNA. So what is the function of the helix 1? This helix 1 makes a contact within the minor groove. Clear? That's mean here the 2 different roles played by the 2 helices. First one, the 1 helix and the 3rd helix. But the second helix role is not said here. The first one that make a contact with the minor groove and the 3rd one that makes a contact with the major groove. So 3rd one, this is called the recognition helix. So this is the HTH motif. And the second one is that HLH motif. That means helix loop helix motif. This is also involved in both the DNA binding and the protein dimerization. Okay, the 2 functions occurred by this HLH, and this is composed of 2 regions of alpha helix and separated by region of variable length and which forms a loop between 2 alpha helices. And length is connecting, the loop varies from 12 to 28 amino acid residues. So here generally 12 to 28 amino acid residues are present within the helices. But here the alpha helix that is separated by variable length, okay, region of variable length, and this form a loop between 2 alpha helices. You can see here, this is the rough diagram of helix loop helix. You can see that it makes a loop, okay, with each other. It is the beta turn which is attached with this one and this one, and this is another beta turn. This is attached with this one and this one. Clear? So they remain in such a way that they form a loop. Okay, and its role is very important because they remain in such a way that they can make a contact with the DNA binding protein very carefully and tightly by making this loop. And this loop is between 2 alpha helices. You can see that first one and second one that attached, that is separated by the beta turn and making a loop with each other. And you can see here this one and this one attached with the beta turn and making a loop with each other. Clear? So this is saying that this loop alpha helix is separated by the region of the variable length and which forms a loop between the 2 alpha helices. So its form generally they remain in an area making a loop. The 2 alpha helix that attach with each other by making a loop, and the number of amino acid residues that varies, that 12 to 28 may be present. Okay, so this is the alpha HLH motif, and this is the H turn H motif. So these are the factors of the, I just have told you, these are the factors that present or seen in the eukaryotic transcription process. So these are there are many factors involved in the eukaryotic process, because eukaryotic transcription, because this transcription process is so complex that many factors must be involved in for doing or continuing the process. So there are also another factors that have already explained in my channel, so you just check out that all. And it is my request to the newcomers, if this channel is helpful to you and if you get the many informations related to the biological topics, so you can obviously subscribe this channel and you just support for the growth of this channel. Okay, and you can comment in the comment section, and please give a like if this video is liked by you. Okay, thank you.